Welcome to today's presentation entitled Laboratory Tests and Biomarkers in Clinical Trials, 10 Things You Should Know Before You Participate. My name is Dr. Gary Zamet, and I will be delivering the presentation to you. I'm the President and CEO of Clinolabs, a full-service contract research organization that provides clinical drug development services to the pharmaceutical industry. I also am a clinical associate professor in the Department of Psychiatry at the Icahn School of Medicine at Mount Sinai in New York. Before we begin, I'd like to thank all of you for taking the time out of your busy schedules to participate in today's session. I hope that you'll find it both interesting and informative, and that it helps you understand the clinical trials process a bit better. There are several learning objectives for today. We'll begin by answering the question, what is a clinical trial? We will then address a topic that is of significant importance to all those who participate in clinical trials, and that is the issue of informed consent. From there, we'll move on to discuss the various types of laboratory tests that commonly are performed in clinical trial settings, including blood tests, urine tests, electrocardiograms or ECGs, and other relevant tests. I will attempt to put these tests into perspective by discussing the way we determine if findings are clinically significant or not clinically significant, and I will relate these to study inclusion and exclusion criteria. In other words, the criteria that determine whether a person is enrolled into a clinical trial or not. We'll speak briefly about biomarkers and how they are used, about how you can gain access to your clinical trial laboratory results, and how the matter of subject safety, that is, your safety, always must be the first concern of the research physician and of all those who participate in clinical trials. So what is a clinical trial? Clinical trials are research studies involving human subjects that serve to test drugs or devices under development. It's through clinical trials that doctors, drug and device companies, and regulatory agencies like the Food and Drug Administration are able to bring new, better, and sometimes life-saving treatments to market. Clinical trials are needed to determine the effectiveness and safety of medications under development, first by testing the drugs in normal healthy people, and later extending testing to people with medical illnesses. There are many steps involved in bringing a new drug from the chemist's bench to the patient's bedside. The most important of these steps is not related to drug discovery or chemistry or manufacturing or sales. It is, in fact, the conduct of clinical trials. According to some industry reports, the pharmaceutical industry spends more than $44 billion a year on research and development. But it is the clinical development costs that represent the lion's share of that expense. More than 60% of those dollars get spent on clinical trials. Clinical trials are conducted in 3 different phases prior to a drug's approval, phases 1, 2, and 3. One additional phase, phase 4, includes studies conducted after marketing approvals, after a drug is approved. Trials performed during the first of these phases of clinical development provide important information that constitutes the manufacturer's evidence for carrying a drug forward, and it forms the basis of the new drug application that is submitted to the FDA. Although these phases are represented in consecutive order here on this slide, you should know that it's common for studies in these phases to overlap or sometimes even run concurrently. So let's break down, though, what these different phases are. Phase 1 studies typically are clinical trials that involve a small number of healthy volunteers. Sometimes patients are involved, but for the most part, it's healthy volunteers. The sample size, or the number of subjects tested in these trials, often is small. It's not unusual for a phase 1 study to include maybe 12 subjects, or 24 subjects. In these trials, we're typically examining what is known as the pharmacokinetics of a drug. How does the drug move through the body? So we want to look at how quickly the drug is absorbed when it reaches its peak levels in the blood, and how quickly it's eliminated through urine or feces. We're also looking at the safety of the drug by asking participants how they feel after they take it. In clinical trials, safety is measured in part by the occurrence of what we call adverse events. So these are typically untoward or negative reactions to a drug. If we administer a drug in phase 1 and there are no negative reactions, it may mean that we can administer higher doses if needed. If we administer a drug in phase 1 and many adverse events are noted, it may mean that we have to use a lower dose, or it may mean that the drug is not suitable for development at all. In any event, phase 1 studies begin to provide the foundation for later trials in a drug development program. Phase 2 studies involve larger samples than phase 1. At this point, that the drug typically is being administered to patients. In fact, administered to hundreds of patients in a clinical trial. These patients are typically those who have been very carefully defined, because they're the first people with a disease or the medical condition for which the drug is going to be intended. So they're very carefully defined. The objective of phase 2 is to determine not only if the drug is safe, but to test if it shows any of the expected therapeutic effects in the patient population that it's intended to serve. The information that we get from phase 2 studies sets the stage for larger phase 3 studies. Phase 3 trials are, as you can imagine, the largest studies that a pharmaceutical company will do. These are decisive in determining the efficacy or effectiveness of a drug and the safety of a new drug. Phase 3 studies typically involve large patient populations, so most commonly thousands of patients, and they may be conducted at multiple investigator sites in multiple countries across the globe. The final results of phase 3 studies will be submitted to regulatory authorities and will provide the required information needed for those authorities, like the FDA, to make a decision regarding the approvability of a drug, whether or not a drug can be approved for marketing. And finally, as I said earlier, phase 4 trials are performed after a drug has been marketed. These trials might provide helpful information regarding the drug or medication that was not collected as part of the first 3 phases. So it might be looking at some added benefit on the efficacy side, or it might be examining more carefully a potential risk on the safety side. Now, let's turn our attention now to informed consent. This really is a critical component of the clinical trials process. Following the research atrocities committed during the Second World War, the Nuremberg Commission established a code in 1947 that specified the way in which research with human subjects could be conducted. The code consists of 10 points. The first and most important of these is that anyone participating in an experiment must give what is known as informed consent. This means that all participants must be informed that they're participating in a research study. They must be given full disclosure about all aspects of the research study, and they must be given the freedom to choose to participate or not. Nobody can be forced to participate in a human experiment. The Nuremberg Code also gives rules for running the experiments. So, for example, participants can leave the experiment at any time. Even though they join a study, they can leave at any time for any reason. Doctors, in turn, must stop the experiment if they realize that it can harm the participants. Also, no experiment can be made where the risks outweigh the benefits that can be had from it. The Nuremberg Code was adapted in the Declaration of Helsinki in 1964 and now represents the standard code of ethics by which all research studies with human subjects are performed. So, human subjects participating in clinical trials have important rights that protect them from the risks or dangers associated with human experimentation. Let's turn our attention to laboratory tests. We'll be talking about blood tests, urine tests, pregnancy tests, urine drug screens, and alcohol breathalyzer or blood tests. We'll be talking about other things as well, but these are some of the main points. So let's begin with blood tests, because in order to participate in a clinical trial, you're most likely to be seen in the office for a screening visit. And at the time of the screening visit, the doctor or nurse will ask you about your health, ask you about your medical history, any symptoms you're experiencing, and then along with that, will take some blood and urine in order to do some routine tests. Well, the complete blood count is one of the typical blood tests that will be performed. The complete blood count consists of the following: a white blood cell count. White blood cells protect the body against infection. If an infection develops, white blood cells attack and destroy the bacteria or virus or other organism causing it. White blood cells are bigger than red blood cells, but fewer in number. When a person has a bacterial infection, the number of white cells rises very quickly. Red blood cell count. Red blood cells carry oxygen from the lungs to the rest of the body. They also carry carbon dioxide back to the lungs so it can be exhaled. If the red blood cell count is too low, an indicator of anemia, the body may not be getting the oxygen it needs. If the count is too high, it's a condition called polycythemia, and there's a chance that red blood cells will clump together and block tiny blood vessels. It may make it harder for red blood cells to carry oxygen. Hematocrit is a test that measures the amount of space red blood cells take up in the blood. The value is given as a percentage of red blood cells in a volume of blood. For example, a hematocrit of 42 means that 42% of the blood volume is made up of red blood cells. Hematocrit and hemoglobin values are the two major tests that show if anemia or polycythemia is present. Hemoglobin. The hemoglobin molecule fills up the red blood cells. It carries oxygen and gives the blood cell its red color. The hemoglobin test measures the amount of hemoglobin in the blood and is a good measure of the blood's ability to carry oxygen throughout the body. Red blood cell indices, there are 3 of them. One is the mean corpuscular volume, the other, the mean corpuscular hemoglobin, and the last, the mean corpuscular hemoglobin concentration. These are all measured by a machine, and their values come from other measurements in a complete blood count. The MCV, or mean corpuscular volume, shows the size of the red blood cells. The mean corpuscular hemoglobin is the amount of hemoglobin in an average red blood cell. And the mean corpuscular hemoglobin concentration measures the concentration of hemoglobin in an average red blood cell. These numbers help the doctor diagnose different types of anemia or other types of blood disorders. The platelet or thrombocyte count is important. Platelets or thrombocytes are the smallest types of blood cells. They're important in blood clotting. When bleeding occurs, it's the platelets that swell up and clump together and form that sticky plug that stops our bleeding. If there are too few platelets, people can bleed too much. If there are too many, there's a chance of a blood clot forming in a blood vessel. Mean platelet volume is the measure of the average amount of platelets. The mean platelet volume is used along with the platelet count to diagnose certain diseases. Now, one component of the One component of the complete blood count is the differential. The differential measures the percentage of each type of white blood cell that you have in your blood. These are neutrophils, lymphocytes, monocytes, eosinophils, basophils, and little baby neutrophils called band neutrophils. We know that there's a standard range or percentage for each of these white blood cell types. For example, neutrophils probably represent 40 to 60% of the white, and lymphocytes maybe 20 to 40%, and the others representing less. But what we understand regarding these values is that too many or too few of the different types of white blood cells can be an indicator of an infection, an allergic reaction, a toxic reaction to medication or to chemicals, or it can be an indicator of certain types of disorders like leukemia. So getting the differential test done helps the doctor also determine the normality of the blood profile. Complementing the complete blood count is something called clinical chemistry. And clinical chemistry is really an additional panel of tests that is performed using blood samples. It gives us information about electrolytes, such as sodium, potassium, and chloride, which are critically important to cellular functioning. So gaining that information about those levels tells us how cells are functioning. It also provides information regarding kidney function, liver function, and heart functions. Overall, these tests provide the physician with insight regarding the presence of health problems or medical disorders in the people being tested. Now, as we look at blood tests, there's no way for me to go into the specifics regarding the meaning of each individual laboratory test and each individual abnormal finding. But it is fair to say that abnormal tests on blood work may be a sign of a health problem or a medical disorder. If you're screening for participation in a clinical trial and you're told that you have abnormal test results, the investigator can provide you with information regarding the abnormal results and the potential meaning of those results for you. However, it's important for you as a clinical trial participant not to stop there. It's always advisable for you as a study participant to review this information with your primary care doctor. It's important to interpret laboratory tests in the context of overall patient care. Therefore, an abnormal finding for one person may not mean exactly the same thing as it does for another. So someone familiar with you and your health and your health care should be involved in interpreting those test results with you. As in so much related to clinical trials, there is always some risk, and blood tests do come with risks. Fortunately, for blood tests, the risks are generally minor. You might experience temporary pain or discomfort from the needle stick. You might experience some bleeding from the place that the needle was inserted. You might experience the feeling of lightheadedness, and some people might actually faint when they get their blood drawn. You might experience some bruising at the place where the needle went in, or possibly some blood might accumulate underneath the skin there. It takes a while for that kind of bruise to go away. Or although it happens rarely, you might get an infection at the place where the skin was punctured. These are risks associated with getting blood drawn. Fortunately, most of them aren't minor, but you should be aware of them. Urinalysis. In association with blood tests in a clinical trial, urinalysis is commonly performed. We typically take approximately 30 to 60 milliliters, that's a few ounces, in a specimen cup, and the urine is examined for color, cloudiness, odor, and clarity. These are inspections done by eye, or in some cases can be done by a machine. Abnormalities in color or clarity, cloudiness, might be suggestive of an infection, could be suggestive of dehydration, it could be suggestive of red blood in the urine, it could be suggestive of liver disease, or even muscle weakness or breakdown. Certain medications also change the color of urine. Very foamy urine may represent large amounts of protein in the urine. All of this analysis is followed by a microscopic analysis. This requires only a very small amount of urine to be placed under a microscope. The cells are examined, the bacteria and the crystals in the urine are examined to provide further clinical clues about bodily functions like kidney functioning, diabetes, hypertension, liver disease, and so on. So the urinalysis is really an important complement to the blood test. Now, for anyone who has participated in a clinical trial, you probably know that the urine drug screen is one component of the screening. The urine drug screen is performed to ensure that subjects have not taken any illicit drugs or any disallowed medications. I've listed a few of the common drugs that are tested in a urine drug screen. These include things like amphetamines, barbiturates, benzodiazepine, cocaine, marijuana, methamphetamine, opiates, phencyclidine. Other drugs can be tested in a urine drug screen. For example, cotinine, which is a byproduct of nicotine, can be tested to determine if you're a smoker. Certain levels of antidepressants can be tested if the study is going to exclude anybody who is taking an antidepressant medication. So the urine drug screen is tailored to the study. There are two primary reasons for urine drug screen. One is to protect the safety of the subject. And again, as I hope I've already intimated in the presentation thus far, safety is the first and primary concern of the physician investigator. Since we're working with investigational or experimental medications, we don't know how those new drugs might interact with other drugs because the studies may not have been done yet. So given that that's an unknown, we don't want to mix these drugs together. Safety is our first concern. Then secondly, the presence of other drugs in the system can cause uncertainty, or in experimental terms, what's known as a confound, can cause a level of uncertainty in the results because we don't know what to attribute the findings to. Is the finding that we get attributable to the investigational medicine, or is it attributable to one of the illicit or disallowed drugs, or is it attributable to an interaction among those drugs? We don't know, so we want to make sure that those drugs are not on board at the time that subjects are screened for participation. There's a couple of important notes. If you are using drugs, the urine drug screen is likely to be positive. We have encountered situations where We ask people on the telephone about their drug use, and then they deny any drug use, and then they come into the office, and we screen them in the office, and the doctor or nurse asks them at the time of the office visit if they're using any illicit or disallowed drugs, and the person will deny using any of those drugs. And then, lo and behold, when the results of the urine drug screen come back, they come back positive for one of the illicit or disallowed drugs. It's really not appropriate to misrepresent that, and also the likelihood is that misrepresentations will be detected. So if you're using drugs, the urine drug screen is likely to be positive. There are certain drugs that can be detected in your urine for up to 12 weeks following your last use, and this can be so no matter what you may have heard regarding ways to foil a drug test, like drinking a lot of water or drinking vinegar or things like that. This really doesn't work. There are some false positives that have been known, though. Antidepressants or cough medicines can result in false positives for amphetamine. Antibiotics can result in false positives for marijuana. The consumption of poppy seeds has long been known to result in a false positive for opiates. So you should know this and have an open dialogue with the physician or nurse investigator when you're going through screening. But by all means, be truthful when asked. Your safety is at risk. Next is the alcohol breathalyzer or blood test. The breathalyzer is identical to the test that is used by police. The breathalyzer units that are used on most clinical investigator sites are calibrated, and they are sensitive to alcohol that has been consumed within the last 24 hours. The only acceptable result of an alcohol breathalyzer test from most clinical trials is going to be 0. That means nothing. As you know, alcohol limits for driving safety are at various levels from state to state. It could be 0.08, it could be 0.10, but in clinical trials, we're really not trying to make a determination if somebody is too inebriated to drive or not. We are excluding people who have been drinking. So the only acceptable result is a 0. A false positive can be obtained due to the consumption of foods or fluids that contain alcohol, like cough syrup or mouthwash, or even if you had one of those Christmas candies that somebody has put a little rum inside the chocolate, it can produce a positive result. So you should be aware of this kind of thing. Okay, I'm going to skip off of blood and urine tests now to talk about electrocardiograms or ECGs. Electrocardiograms are tests that represent measures of heart electrical activity. The normal heart, everyone's heart, produces electrical signals. These signals originate from a part of the heart called the sinus node, and they travel from the top to the bottom of the heart. As these electrical signals travel, they cause the heart chambers to contract, thereby pumping blood throughout the body. The electrocardiogram shows how fast your heart is beating. It shows how regularly your heart is beating. And if you have a regular rhythm or if you have an arrhythmia, an irregular rhythm, and it also shows the strength and the timing of the electrical signals that are causing your heart to pump. The electrocardiogram can be used to assess heart health and safety and can be an indicator of one's risk of heart attack, of heart failure, or of other heart disorders. So the electrocardiogram is typically an important part of the screening visit for clinical trials. One thing that you may like to know is that electrocardiograms are simple and painless. This image of a gentleman getting his EKG done shows the typical lead hookup for an electrocardiogram. And you can see the blue electrodes or sensors that have been placed over the left side of his chest, kind of below and in a semicircle around his left breast. There are two leads that are placed right around the middle of the chest, and then there are four leads that you don't see placed at his wrists and at his ankles. And based on those leads, we're able to collect multiple leads of electrocardiographic activity. The typical electrocardiogram will report 12 leads or 12 channels of activity, and you can see what that typical waveform looks like in the graphic represented on this slide. So the sensors go on with a little bit of sticky adhesive tape. For the most part, men do not have to have their hair shaved. Sometimes, for very hairy people, shaving might be a request, but for the most part, shaving is not needed. For women, in many cases, we don't even have to remove the lady's brassiere. So if women are concerned about that, there are ways to get around that. And the EKG can be simply taken. It's a painless process and one that is completed in just a few minutes. Now, what I've just explained to you regarding the blood tests, QBC, DIF, and urine analysis, drug screening, and EKG, these are all very, very standard tests done at screening and probably at follow-up in clinical trial settings. Within recent years, there's been another test that has become increasingly common, and that is pharmacogenetic or pharmacogenomic tests. These are blood samples that are obtained for the purposes of genetic testing. And it's typically that the investigator or experimenter wants to determine how a person's genetic makeup might influence his or her response to a particular drug. If you've ever heard the popular term personalized medicine, which is more and more popularly used these days, this is the idea behind it, that in the future we'll be able to do a genetic test and then determine what medication a person is most likely to respond to, and then prescribe that medication straight away, rather than trying different medications at random to see which one a person may do better on versus not. So the pharmacogenomic testing is really a very forward-looking type of test that investigators are trying to do to help us understand better ways to prescribe in the future. Because it's new, sometimes an independent informed consent form will be required, and maybe not just because it's new, because it is a type of information that is highly personal. Your DNA is the only thing that makes you truly unique, and some people don't want to have their DNA analyzed. So sometimes the pharmacogenomic testing is included in the standard consent form that you sign and agree to do, or it may be a separate document that you sign and agree to do. If you don't want to have your DNA tested, in most studies, the pharmacogenomic testing is optional, and you can decline to do it even though you participate in the rest of the trial. Other tests may be performed as part of a clinical trial. And there are literally dozens, if not hundreds of them, that can be ordered as part of the trial. these are just a few examples. If you were going to participate in a diabetes study, your HbA1c might be measured at the screening visit and then at later visits. This particular value tells you how well your sugar has been controlled over the past several weeks or months. If you were going to participate in an infectious disease study, such as a study for HIV-positive people, the screening test might involve assessment of HIV antibodies. If you were going to do a cancer study, a prostate study, prostate-specific antigen or PSA tests might be done, or the cancer antigen 125 might be done if you were going to join an ovarian cancer study. If you were going into a reproductive safety study and you're a male, the investigator might request that you provide sperm samples so that they can assess sperm count, sperm morphology, and sperm motility. How many, what do they look like, and how much do they move around? So there are just dozens and dozens of additional tests that may be required. I think the important thing for you as clinical trial participants to know is that all of these tests should be specified in the informed consent form. You should be aware of every type of test that's going to be performed. And of course, by providing informed consent, that is reading the consent form, discussing the study with the investigator, and signing the consent form, you're agreeing to have all of those tests done, and you're aware that they're being done. It's worth mentioning that the types of tests that will be performed at your screening visit are often the same types of tests that would be performed at follow-up visits. However, they may be used in somewhat different ways. At the screening visit, tests are used to determine if you're eligible for study participation. For example, if you were a normal healthy volunteer who was planning to participate in a study, we would look for laboratory tests that generally fall within the normal range. If, similarly, you were planning to participate in a study of high cholesterol and you were a patient who had high cholesterol, we might be looking for a specific panel of tests to indicate that you meet the criteria for high cholesterol in the study. Once clinical trial participants are enrolled in a study, follow-up tests may be done to confirm eligibility for participation. These follow-up tests may be done prior to receiving investigational drug or any type of comparison medication. But follow-up tests also may be done to assess subject safety. We want to make sure that the drugs being tested don't result in significant laboratory abnormalities. And finally, we want to determine the potential overall impact of a new drug on laboratory values. Some lab values may change as a result of clinical trial participation. If these values are not considered to be clinically significant, maybe it's okay for the subject to continue in the trial. If the physician investigator believes that the change in laboratory values represents a health risk to the subject, the investigator may terminate that subject's participation in the trial. So we use the screening visit and follow-up laboratory data in similar but slightly different ways. As a clinical trial participant, you may hear the terms clinically significant versus not clinically significant. So let's discuss the concepts of these. A clinically significant finding is one that is considered to be of real practical importance to the subject's health and well-being. For example, if someone was screened to participate in a clinical trial and they consistently showed a blood pressure of 180 over 110 or greater, it is certainly going to be considered clinically significant. In fact, a blood pressure of 180 over 110 would indicate that emergency care is needed. That candidate would be referred to urgently to an emergency room. Under these circumstances, obviously, clinical trial participation is not considered appropriate. A not clinically significant finding is one that is not likely to be important or of immediate importance to the subject's health and well-being. So back to our blood pressure example, if someone had a blood pressure of, say, 125 over 85, 130 over 88, it might be a blood pressure that the doctor would be interested in watching over time, but not one that would warrant immediate treatment or exclusion from a clinical trial. Again, as within all matters related to clinical trials, the safety and well-being of the subject is the investigator's first concern. With all of the lab values, the investigator will look at every blood value, every urine value, every drug screen value, every EKG, and it's the investigator's obligation to mark those clinically significant or not clinically significant. Excuse me. So, as a trial participant, you can be assured that every value is going to be checked and double-checked. Lab values relate to study inclusion and exclusion criteria. In general terms, every clinical trial has inclusion and exclusion criteria. In other words, there are inclusion criteria for things that you must have in order to be included in a study. And there are exclusion criteria that indicate what you must not have in order to be included, in other words, if you meet an exclusion criterion, you can be enrolled. In practical terms, let's pick an example. If someone wanted to be involved in a study of people with depression, say, they might have to be, on the inclusion side, they might have to be between the age of 18 and 64, male or female, have to meet the current diagnostic standards for major depression. They may have had to have symptoms for a minimum of 3 months or longer, and they may have to show specifically some failure to respond to currently available antidepressants or something like that. So these are the things the person must have. On the exclusion side, the things the person must not have, the person must not have an unstable medical condition, a history of uncontrolled high blood pressure, clinically significant abnormal blood test values, clinically significant abnormal urinalysis, use of illicit drugs or disallowed medications reported by self-report, or use of illicit drugs or medications indicated by urine drug screen, abnormal EKG, etc. So if you have any of those things, you can't be enrolled. Excuse me. The criteria for inclusion and exclusion are very, very strict in clinical trials, and that is because if the investigator or anyone else in the clinical trials process doesn't adhere to them very carefully, it's considered what's called a protocol violation, and it may make the information completely unusable right from the get-go. Excuse me. Okay, we'll talk briefly about biomarkers. I promised you at the beginning of this presentation that I'd be talking about biomarkers and what they are. But biomarkers are any measurable characteristic or test that reflects the severity of a disease or the presence of a disease, or is an indicator of the physical state of the subject. Biomarkers can be used for early diagnosis, they can be used for drug target identification, and they can be used to evaluate drug response. Biomarkers are being increasingly used today in clinical trials to provide additional endpoints or additional outcomes that can be used to determine whether or not a drug is doing what we think the drug should be doing, or if it seems like the drug would promise to do what we think it should promise to do. Some of these biomarkers are taken in normal, healthy people. Some of these biomarkers are taken in normal, healthy people. Others are taken in patient populations. So, for example, if you were going into a biomarker study for cholesterol, in which a drug is thought to lower cholesterol, then LDL would be a biomarker test that could be done. C-reactive protein, we know, is a biomarker for inflammation. So if we were looking at inflammatory diseases like rheumatoid arthritis, we might look at C-reactive protein as a biomarker. Quantitative EEG, or electroencephalography, which is a measure of brain electrical activity. That might be used to see if a sedative is actually getting into the brain and whether it is producing any changes in brain electrical activity that seem to be consistent with sedation. MMP is a biomarker for breast cancer. And in other cases, cerebrospinal fluid can be used to provide biomarker indications in various types of disorders, including many studies now with Alzheimer's disease. As many of you know, cerebrospinal fluid is a clear fluid that bathes and cushions the brain and the spinal cord, and this fluid can be sampled in tiny, tiny amounts using a lumbar puncture or what sometimes is called a spinal tap that allows us to take just a little bit out and then measure levels of chemicals that are in that cerebrospinal fluid. In Alzheimer's disease, for example, we use cerebrospinal fluid to measure the presence of proteins called tau and beta amyloid that we believe are linked to Alzheimer's disease. As I've stressed throughout the presentation, subject safety is the clinical investigator's first concern. With any lab test or any clinical trial, the physician must gauge the risk of that trial, and you need to be fully informed. You should be fully informed of all of the procedures that will be done. And similarly, if you have any clinically significant laboratory screening tests that perhaps impact your ability to enroll in a trial, you should be informed, and the physician, the investigator, can tell you what that abnormality was, what it means, and encourage you to speak to your primary care doctor about what those findings are. Similarly, if you have abnormal tests during the course of your participation, you have the right to know what those abnormal laboratory tests are. In fact, you do have access to your test results. Your clinical trial source documents are medical records, and those medical records are yours. These include things like physical exams, lab tests, ECGs, any other special assessments that were done, imaging, biomarkers, biopsies, etc. So unless your informed consent form specifically excludes information that you wouldn't have access to, your source documents are medical records, and you do have access to those medical records. If you're interested in more information about laboratory testing in clinical trials or biomarkers in clinical trials, I encourage you to visit the following websites. I did an internet search myself and found these websites to be of help. One is medicinenet.com for lab and diagnostic tests. Wikipedia has a real nice description of biomarkers. If you're interested in general about clinical trials information for the public. CISCRP.org is excellent. And if you're interested in a directory of clinical trials nationwide, it's clinicaltrials.gov. If you're interested in information about clinical trials being conducted at CliniLabs, you can get that information by contacting us by telephone at 212-994-4567. You can visit our website at clinilabs.com/volunteer. Send an email to participate@clinilabs.com. Text volunteer to 313131, or stop by and visit us at our clinic in New York or New Jersey between the hours of 9 a.m. to 5 p.m. Monday through Friday. So with that, I will open up the floor for questions. All right, first question coming in is, I was asked to participate in a clinical trial that involved frequent blood samples. How much blood can I give for it to still be safe? Well, all right, so this is a good question. There definitely is a limit on how much blood can be drawn in a clinical trial setting. Many institutions will limit the total amount of blood to be drawn at any one time to something around 200 milliliters of blood, and this depends on a person's body weight. The heavier you are, the greater the blood volume you have and the more blood you can actually give. But even though there's a limit on how much you can give at one time, there's also a limit in the total amount that can be drawn in any period, and this can vary from one organization to the next. Given the fact that any study that you participate in will be reviewed by an institutional review board or an ethics committee, you can be assured that the amount of blood to be drawn in that study will have been considered by that ethics committee for that trial. But if you have concerns about the total amount, then you certainly can speak to the investigator and ask the investigator what to do, or what the total amount to be drawn may mean for you. Okay, sorry I got distracted. There are many questions coming in. Next, I have, what is the acceptable range for normal blood pressure for clinical trial participation? Well, the acceptable ranges vary from protocol to protocol. If we look at the ranges set forth by the American Heart Association, we know that normal is anything less than 120/80. That's the American Heart Association rule. If you're 120 to 139/80 to 89, that's kind of a state of pre-hypertension. If you are above that range, you fall into various levels of hypertension, stage 1 or stage 2 hypertension, or excuse me, as I mentioned earlier, if you're 180/110, then it's considered a hypertensive crisis and you're in need of emergency care. One thing to keep in mind is that one blood pressure measurement does not constitute whether or not you're hypertensive or not, or whether you have high blood pressure or not. It's usually a series of multiple readings over time. So, if your blood pressure is a little high on one day, it doesn't necessarily mean that you have high blood pressure or that you'd be excluded from a trial. The key is to work with that, physician investigator to know. One question that just came in, which is, does smoking exclude you from most trials? Again, that's a good question. My experience is that smoking is more often identified as an exclusion criterion for various trials. So if you are a smoker or a heavy smoker, you may be excluded. Some protocols will not comment on smoking. Others may allow light smoking, which they consider up to 5 cigarettes a day or so, and then others want smokers totally excluded. And if the sponsors are very concerned about excluding smokers and they don't want to rely exclusively on a person's self-report, they will do a cotinine test to determine if a person has been smoking. Cotinine tests are positive in smokers. They're not positive in other people. They're not positive in secondhand smokers, et cetera. So it all depends, and if it's something that is of concern to you, when you do a phone screening, that would be the time to ask if a smoker would be excluded. One other thing to note is that we have recently been running trials for smokers in smoking cessation studies. And of course, in those cases, we're looking for people who are avid smokers. So what might exclude you in one study might mean that you're included in another. Okay. Let's see. This question of why is participation in some trials labeled as a number rather than by name and the results not available to the individual? One of the things that we do in general in a clinical trial is to limit the risk of exposure of personal health information. So the subjects who are participating in the trial are not identified by name. They are instead identified by number. So unless you're the investigator or a member of the investigator's staff, like the research coordinator, and you have access to information about the subject's identity, if you were looking at the medical record, otherwise you wouldn't have access to that person's identity. So everything is de-identified, and it's done so with a study number. And with regard to some results not being available to the individual, to my knowledge, your source document, your medical record is available to you. So results of tests, EKG, blood tests, urine tests, your medical history, all that's available to you. But what is not available is the results of the drug testing. That's something that the investigator or the pharmaceutical company sponsor, they keep that confidential until that they release that information to the general public. And in some cases, people people can even gain access to their results afterward if they continue to have communication with the investigator, and the investigator can get the information from the pharmaceutical company. Sometimes we can find out, what drug the person was on, and give that person feedback, so that if there's a similar drug that's already marketed, or the person wants to wait for the drug to be marketed itself, if it gets approved so they can take it, they can get a hold of that kind of information. Here's the question, why do we fast before trials? What does it change in our labs? So there are a variety of labs that can be impacted by the food that you eat. For example, an easy one would be your blood glucose levels. If you're doing any kind of measure of blood glucose, then if you eat any food, your glucose levels will be higher, or insulin levels will be higher, and so on. So fasting creates kind of a blank slate on which they can do the test. In addition to that, medicines are absorbed at different rates depending on whether or not a person has eaten or not. It's not true of every medicine, but it's true of many medicines. So there are what are called food effects, and the food effects can have an impact on how much of the drug will get absorbed, what peak level it will reach in your blood, and so on. So especially on days when you might be receiving a medication before testing being done, the fasting condition is always desirable. Excuse me. Okay. Okay, and here's another one. If this is a long question, but it seems like I gave blood a few months ago, and it took so long to get the results, and I was told by one of your associates, so one of my associates, that they were looking for something specific and was told everything was okay. With emphasis, just out of curiosity, what were they looking for? This was, okay, so in this particular study, which I'm aware of, we were looking for a particular biomarker in the blood that is a biomarker for Alzheimer's disease. And in this particular study, that finding remains confidential. The biomarker that we're looking for in the blood, unfortunately, was and is, it's a very specialized biomarker. There are only a couple of laboratories that can actually do the testing for this biomarker. And we did have several people who were participating in this study who had blood drawn for the biomarker, but we couldn't give the results because the laboratory paused the analyses that they were doing. I think they were on hold for maybe 3 to 4 months, and we couldn't get the results from the laboratory, even though the laboratory had the blood samples and had them frozen and waiting for analysis. They didn't run the analysis. So that's why there was a delay. But what they were looking for in that particular case was a biomarker for Alzheimer's disease. Okay, and let me see. Okay, and here's another question. Sometimes people worry about reporting their adverse events. Can you be disqualified from later studies from having an adverse event? Well, that's a great question, and I'll try to answer this in the best way I can. So an adverse event, as we had discussed it earlier, is an untoward or negative reaction to study participation, perhaps to the study medication that's being administered. And if you have a negative reaction and you report it, can you then be excluded from further participation in other studies because you reported a negative reaction in one study? I will say to you, in general, I think the answer to that is no. One of the reasons that we do these studies is to detect adverse events because we're concerned about the safety of these investigational drugs. So we want people to report whenever they're experiencing them. And you should not be penalized for having made such reports. However, that said, remember, we're always concerned about the safety of clinical trial participants. So if I was doing a trial and I administered a medication to a subject and their blood pressure shot up, and it shot up so high that they needed immediate emergency room care, and then they came back and they were fine after that, no problem, everything looked fine. The next trial that I would do with a similar type of drug would be a situation I'd be definitely concerned about that patient's safety, and I'd want to make sure that that patient's safety came first in enrolling that patient into the next trial. So I guess my comment to you is, it shouldn't impact your future trial participation, but do keep in mind that the investigator is always going to be concerned about your safety. If it seems like you might be put at risk by an adverse event, that's going to be a concern. Okay, I noticed that we're over. It's after 8, so I think I'm out of time for questions and answers. But I do want to thank all of you who participated. It's truly been a pleasure to speak with you tonight. I appreciate very much the questions that you've posed, and I hope that you found tonight's presentations to be both informative and enjoyable. So thank you all very much